Hi, I'm Jesus Peña. I'm a graduate student at UC Riverside in the microbiology program. I work in the STICH lab where we're interested in the genomes of fungi. My work specifically focuses on the developmental biology of fungi, and one of the ways that I go about studying developmental processes is by extracting their RNA. RNA tells us what gene is being activated or expressed at a particular stage. So we want to isolate the RNA, we want to sequence it, and then we want to find out what those genes are. So for today, I'm actually going to walk you through my preferred protocol for isolating RNA using the Trizol reagent. So let's get started. Okay, so let's talk a little bit about the materials that we're going to be using for this protocol. The very first thing I'm going to point out is this RNase ZAP, which is a chemical that's going to inactivate RNases. We don't want RNases on our equipment or really anywhere near our reaction because RNases are going to destroy the RNA and make it impossible to isolate, or if you do isolate it, you're going to not be able to use it for any downstream application. So RNase will be used to spray down all of the equipment that we'll be using. Then we're going to use a mortar and pestle, which we're going to chill with liquid nitrogen, which is down there, and I won't show you that. We'll chill it with liquid nitrogen, we'll add our tissue, we'll grind it up, and then we'll transfer that to a microcentrifuge tube, like this one, which will contain 1 milliliter of the Trizol reagent. And we're going to keep that on ice for now. And this is the Trizol reagent, which will actually do the bulk of the work pretty early on in the protocol, and you'll see as we proceed. We also have chloroform back here, which will be important for taking proteins out of the solution. Of course, we have ice because we want to keep everything on ice. We want to keep everything cold in order to prolong the stability of the RNA. We've got 70% ethanol, which is going to be used to wash the RNA in a later step, as well as isopropanol, which is going to be used to actually pull the RNA out of solution. And then lastly, we've got this water that's been treated with DPC, which is a chemical which inactivates RNases. With that, we will start the process of chilling the mortar and pestle. So I've sprayed down our mortar and pestle with RNase ZAP, as well as this spatula here, which we're actually going to use to transfer that frozen tissue into the Trizol tube. Okay, so now we're going to chill the mortar and pestle by adding liquid nitrogen. I've already added some off camera to speed up the cooling process, but just to demonstrate, so while the mortar and pestle are chilling, I'm going to set it aside, and then we're going to add our Trizol to our reaction tube. So I like to hold the pipette tip over the source of the Trizol in case it drips. It'll drip right back into the original container. And then we transfer and eject. And as you can see, Trizol is this pink liquid. So we put that back on ice. We cap the Trizol. We throw the pipette tip away in a Trizol waste container because Trizol contains some pretty nasty chemicals. We don't want it to mix in with the regular trash. And by this point, the mortar and pestle will be pretty cold, as you can hear, maybe from the gloves kind of sticking to it. So we'll put the Trizol away, and then we'll get back to homogenizing our fungal tissue. So just before adding the tissue that we're going to homogenize, we're going to add a little more liquid nitrogen. Just like that. And now, depending on the tissue that you're working with, sometimes you have to scrape it off the plate. In this case, I've picked it off the substrate using sterile tweezers. So it makes our job a little easier. We can just pour it right in there. As you can see, I'm wearing a cloth glove on this hand because the mortar and pestle is actually very cold and could probably give you frostbite if you held onto it. So you give a few initial crushes like this, and then that, and you can see it's turned into a fine black powder. So I'm continuing to pulverize some of this tissue just to make sure that we have a very fine powder. So I've been doing this for about 2 minutes now, off camera. Now we're going to take this spatula, which is pre-treated with RNase Zap. We're going to take the spatula, we're going to cool it in liquid nitrogen so that we can start scraping off the tissue from the mortar and pestle to transfer it to our Trizol. So we'll set that aside. Cold spatula, and just very simply scrape the sides. And you can see, kind of, I guess. We'll open up the tube with the Trizol. You'll notice that I took it out of the ice. It just makes it easier to transfer it this way. Take a little scoop. And it's okay if you, if not all of it makes it in. I mean, you want the majority of the sample to make it into the tube, but a little loss is okay. All right, and that's what it looks like at this stage. Inverted the sample tube a little bit. Now it's no longer pink, it's mostly black, which is the color of the fungus that we were extracting from. We'll invert it just a little bit to make sure that the Trizol is mixing, and then put it back on the ice, and we'll wait 5 minutes before proceeding on to the next step. So the next step is to add chloroform to our sample, and this is going to separate proteins from the DNA and the RNA that are present in the sample. I won't actually do this here since I have to do it in a safety hood because of fumes, but I'll demonstrate some of the hand positions that I'm going to be doing as I take out some of this chloroform and add it to the tube. So once we open this up in a safety hood, I like to tilt it slightly so that I'm not pipetting directly over it. That increases the chance of something falling in, something that we don't necessarily want, that would contaminate the stock solution. So we tilt it a little bit, you insert the pipette, and then you draw up the liquid that you need, keeping it over the opening, just as we did with the Trizol, and then taking your sample tube, bringing it near, and then just adding it. So it's that simple. We just want to take every precaution possible to not contaminate our stock solutions. So I'll go do that, and then I'll meet you back here in another 2 to 3 minutes. All right, so now that we've added the chloroform to our samples, we're going to centrifuge it for 15 minutes at 12,000 Gs. We're also going to do this in the cold room centrifuge so that our sample stays cold, since we can't spin it while it's in the ice. We want the environment around it to be cold in order to, again, preserve the stability of the RNA that we're going to be isolating. So we're going to spin our sample at 12,000 G for 15 minutes. Okay, welcome back. So I've gone ahead and taken our samples out of the centrifuge in the cold room. So if you recall, the samples were these like black, sort of liquidy, gooey stuff. Before we even added the tissue, it was actually pink. And after it's been spun for 15 minutes at 12,000 G, it'll come out looking like this. So those are the 3 layers that Trizol, the Trizol reagent, is named for. So this very top layer is our aqueous layer, and that's where the RNA is going to be dissolved. there's this very thin interphase layer between the aqueous layer and the red layer. That is where the DNA has ended up. So Trizol extracts the RNA, the DNA, and the proteins. So that means that this red layer or pink layer, where this like debris is living, that's where all of the proteins have ended up. Because we're interested in only the RNA, we're going to transfer this aqueous layer to a new tube. I like to use a P1000 for transferring the aqueous layer from the original tube to a new tube. Again, we're going to use these filter tips. I'm going to go ahead and open up our new tube down here. You can't see it because it's on the bench. So when we transfer the aqueous layer, we want to keep our tube angled. The pipette goes in, and then very slowly you'll draw up some of the liquid. You want to be very careful that you're not getting any of the pink protein layer or any of the interphase. And we transfer, just like that. And here's presumably where all of our RNA is for that particular sample. Now that we have the RNA isolated more or less, we need to clean it up. There's not just RNA in that aqueous layer. So the first thing I'm going to do is add a little bit of this isopropanol. So the isopropanol is going to precipitate the RNA. It's going to draw the RNA out of solution. And then we want to change this to 500, so we're going to be adding 500 microliters of isopropanol to each sample. You'll notice that we keep it in foil because it is light sensitive. Once we've added the isopropanol, we're actually going to let it incubate for about 10 minutes. Prior to starting the incubation, I like to very gently invert the tubes just to make sure that that isopropanol is actually mixing with all of the contents. And you'll see that as you mix it, it becomes cloudier, which is usually a good sign. It usually means that the RNA is coming out of solution. So again, here's the before, and now we'll invert it a little bit. Let me take some of that condensation off. And now you can see it's very cloudy with RNA. So we'll set our timer for 10 minutes and we'll come back. So the next step is going to be to take our tubes back into the cold room, back into that centrifuge, and this time spin it for 10 minutes instead of 15 at 12,000 G. So I'll see you in another 10 minutes. So our samples actually look really great. Well, okay, that's relative. Let's not get ahead of ourselves. So as you can see here, we actually got a little bit of a pellet. And the reason that we can see it, or the reason I think we can see it, is because the fungus that we extracted this RNA from is pigmented. So it's a melanated fungus. So some of that pigment is going to be left over. So for this next part, we have to get rid of that isopropanol. I like to use the P1000 to start. So again, very carefully, we don't want to disturb that pellet, and thankfully we can see it, so we know that we will or should be able to avoid it. Waste container. So now we have an RNA pellet. The next step is to wash it with 70% ethanol, which we keep in our -20 so that it stays cold. All right, so here's our ethanol, our cold ethanol. And we're going to add 500 microliters to each tube. The reason we're adding ethanol is to get rid of some of the salt. So this is a wash step, and we're quite literally washing the RNA pellet. So again, just very gentle inversions. The manufacturer's protocol says to resuspend the pellet, but I like to be as gentle as possible just to protect it a little bit. It's a fragile molecule. It's a lot of fragile molecules, and we want to keep them as intact as possible for whatever downstream application. In my case, it would be sequencing the RNA. One thing I like to do is also to turn it on its side like this and make sure that ethanol touches the walls, because there's going to be some leftover salts that are just clinging to the side of the tube as well. Okay, and now we centrifuge for the last time in this protocol. This one is actually shorter, so it's a 5-minute centrifuge at 7500 G. So I will see you in 5 minutes. All right, so our samples have been spun for 5 minutes at 75 G, or 7500 G more precisely. The next step is to get rid of that ethanol. All we want left is that very nice RNA pellet. So in order to get rid of the ethanol, what I'm going to do is use this P1000. I've set it to 500 so that we can get the exact volume back out. Very carefully, without disturbing that RNA pellet, just insert, make sure that you're aware of where the pipette tip actually is. Get as much of that out as possible. Okay. So now we're going to take the tubes over to our heat block, which is on the other side of the lab, and I'm going to leave it in the heat for about 2 to 3 minutes just to air dry it. So now that our pellet is dry, we're going to resuspend it or redissolve it in this DPC treated water, DEPC treated water. It's nuclease free, so there aren't any DNases or RNases, and that's because of this chemical DEPC, which clears them out. So actually, I'm going to use a P200 this time to resuspend it. So I like to use a smaller volume to resuspend the pellet so that it's a little more concentrated. All right, and one of the ways that we get this pellet to dissolve is by just using this tube rack and kind of running the tubes along, so you'll hear that sound. In some cases, we'll want to put it back on a heat block, which will help dissolve the pellet, but I don't think it's necessary in this case because most of it is already dissolved. So that concludes the RNA isolation protocol using the Trizol reagent.